Glycoproteins, as their name implies, there's actually three classes of it. The first class is literally referred to as glycoproteins. The second class is known as proteoglycans, and then the third class are known as mucins. There's the majority of it, and then there's the minority of it. Minority. So the majority of a glycoprotein is a protein. That's the majority of it. The minority of a glycoprotein is the actual sugar parts of it. Now, glycoproteins can exist usually as a part of a soluble system, but also can be embedded on the cell membrane surface. So the protein tends to usually be soluble, but colloquially we can kind of refer to this as other things. So what would be an example of a soluble effector molecule or a soluble glycoprotein? Any type of a hormone that is not a steroid hormone arguably is a glycoprotein hormone. We're going to talk about one called EPO later on, but other things as well exist as glycoprotein hormones. So hormones would be an example of a glycoprotein. Next we have are the proteoglycans, and as their name implies, proteo meaning protein and then glycan meaning sugar, doesn't really say too much. Let's talk about the major and then the minor substituents of this. So the majority of a proteoglycan is actually the sugar part. And then the minority of it is the protein. The type of sugar that we're talking about in a proteoglycan is known as and this is a long word, glycosaminoglycan. Glyco meaning sugar, samino, amino, think about amide, and then glycan implying just another sugar. These tend to be disaccharide repeats, meaning they're just two in tandem back and forth. Two examples that this can get from this ways we can comment this glucosamine and then galactosamine. So you know those morons that take those stupid supplements thinking that it's going to help their joints because people like to sell them off. It's not going to help your joints. It doesn't get absorbed into the tissue spaces here. But anyways, galactosamine is the other one. And a structural feature of these disaccharide joints would be that they all have carboxylate or their sulfates attached to them. So what would be the function of these type of molecules? So these really play a role in movement, but I like to think of them as being parts of the joints. These play a role in cushioning things. So I'm going to say shock absorption. Whenever you walk on your feet or whenever you move anywhere, you're using your proteoglycans to help you move. Lastly, there are the mucins, and just like before, they're similar to the proteoglycans in that the major component of them is the sugar and the minor component of them is the protein. So for the mucins, the sugar that's involved in this is known as N-acetylgalactosamine. Yeah, think about that. Acetylgalactosamine. Galactose amine. Hence the N there. So this is an amino sugar, obviously, because I said that it ends in an amine and it's an N. And so one of the things that are, I guess, some structures about this sugar as a whole that make it really good are really just the structure of the mucin. Let's see the whole structure of the mucin. That make it pretty unique. They have cysteine-rich regions. They have something known as variable number of tandem repeats, which are going to be really rich in serine and threonine residues. And so we abbreviate that as VNTR. Variable number of tandem repeats. These tend to have a lot of serine and a lot of threonine, and I'll explain why serine and threonine play a role in a lot of this after that. So in case you didn't know what mucin did, and I don't really like talking about it because it fucking grosses me out, but this is along the lines of mucus. That's where it comes from. Where does it come from in your cells? Goblet cells of your respiratory tract, your urogenital tract, really. And that's the function of those. So the last thing I wanted to mention with glycoproteins would be the ways in which we can link things together, the ways that we connect the protein with the sugar. So there's two forms of linkage that we know of, at least, that we've been able to identify. There's N linkage like amide linkage, and then the other one is known as O linkage. In case you don't remember, N, we're talking about amide for that. For the N linkage, the main, I guess, molecule, it's kind of the name of the game here. I'm just going to go ahead and draw these two kind of branching off like this, and then I guess in the center we can draw the main molecule that we're talking about, but asparagine, that's the name of the game when it comes to forming an N linkage. So one of the things that all N-linkage proteins have in common is this pentasaccharide core that's going to consist of 3 mannoses, 6 carbon sugar, which is what mannose is, and then 2 N-acetyl glucosamines. So pentasaccharide core. Remember, penta meaning 5, so 5 sugar core, right? What does this pentasaccharide core consist of? You're going to have 3 of those are going to be mannose, and then 2 of those are going to be N-acetyl glucosamines. Ain't that a mouthful? But anyways, 3 plus 2 gives us 5. And just for fun facts, in case you don't remember, mannose is a 6 carbon sugar. I really want to reiterate the asparagine formation of the N-glycosidic linkage. So what you have in this, if you remember the structure of asparagine, it's a really simple amino acid. I'm just going to draw it, ignoring the other side groups, and just draw the R group of it. But the R group of asparagine is just a CH2 and then an amide. So in asparagine, or in linkages with asparagine, this amide here is going to act as the nucleophile that is, and I'll switch colors just to really, I guess, drive home my point here. This is going to be the part that is the nucleophile that's going to bind to the sugar in some way. And so that's how we're able to form these bonds. In case that wasn't obvious, it's going to act as a nucleophile to attack the carbonyl carbon part of your sugar. In this case, I'm drawing it as an acetyl group because it classifies it as an acetyl glucosamine, or as an amide linkage. The other part of it, though, and it doesn't really, the textbook didn't go into a whole lot of details of explaining what it does, but there's this oligosaccharide patterns that exist as well. And this right here, this pattern is going to vary from structure to structure. Okay, so let's talk about the O linkage, which is pretty straightforward. O linkage, just like we've talked about for always, this is where they're attached to the OH group of the amino acid, the R groups. In this context, the 2 R groups, or the 2 hydroxylated amino acids that we're attaching ourselves to, are serine and threonine. And I'm going to make the assumption that tyrosine just wouldn't work because it's so damn big. But they didn't say why tyrosine wasn't used. But in either one of these, I'll just draw blue. I'll show serine as an example. You can do the same thing with threonine. Serine is really basic, simplistic amino acid there. So with serine, we have a hydroxyl group with lone pairs on it. The lone pairs, as you can probably imagine, are going to act as a nucleophile that's going to attack the sugar and then bind the 2 things here. So this is how we link proteins to sugars, through N linkages using asparagine, and then through O linkages using serine and, I didn't draw threonine out there, but threonine as well. Tyrosine, for whatever reason, is not included in this group, and neither is glutamine for whatever reason. But anyways,